everyone. It's such a pleasure to be here. And I have to admit, I've never been to Ireland before. And I don't know if I've ever been more impressed to get to go to a new country. It's just been so wonderful to be here and see the countryside and be here with you and see some people I've seen by email many times. Like Sylvie has helped me many times, and Kevin and Hans, all of you. Thank you so much for having me today. So what I would like to do is something a little bit more general than what I talked about in what the title in the abstract. And that is to give you kind of an overall picture of how science has changed and how our view of pathways to biological crystallization or crystallization of minerals in the presence of water has changed dramatically over the last few decades, if I could say that. So let's get started. The first thing I want to do is just set up a picture of the landscape over which we've come. So from the 1800s, or from way back, from the beginning of time, people have been fascinated by biomineral structures and the idea that we can form crystals in our tissues to form all of these functional materials. And so at the very beginning, of course, there were no analytical techniques to speak of. And so everyone was making these elaborate drawings, and the libraries, especially here in Europe, are filled with these fantastical illustrations of what biominerals and organisms in general look like. And so when I look at something like this, I think of Dr. Hazen's presentation on Tuesday, where he said each specimen is a time capsule waiting to be opened and read. And that's exactly what we've been doing ever since the very beginning of drawing these illustrations, is opening these time capsules. So let's take a look at that. As we go into the 1900s, different groups and parallel efforts around the different continents were trying to get a handle on what these things are made of. And here you see three independent treatises where happening in Germany and Washington and Moscow, where they were trying to analyze the composition of different marine organisms and tabulating data. And we've actually looked at some of these from some of our own research early on. And so here's just yet another way to look at these. This is a time capsule, if you will. And then if we're going into the 1960s, then we have around this time the advent of X-ray diffraction. And here's an extraordinary letter written in 1961, where Heinz Lowenstein at Caltech, who's one of the early people interested in the chemistry of biominerals, sent these chiton teeth as a mollusk, sent these teeth to Julian Goldsmith in Chicago, and he X-rayed them on this X-ray diffractometer and determined that they were made of magnetite. Just amazing old letter from 1961. And so from there, we see that we have actual minerals and what start to characterize what they are crystallographically. In the 1970s, around this time, was the emergence of scanning electron microscopy. And there were explosions of papers like this one that made the cover of Science magazine, showing and I'm trying to get used to my pointer here, showing the concept of how nacre, that material that we think of as is used as a semi-precious for stones for jewelry and other things, nacre having diffracting light, really because the aragonite grows by this birth and spread mechanism over in layers and layers, and it's the spacing of these layers that determines the way the light breaks up and gives it its beautiful iridescence. And so we start to really get an idea of what the microstructures are of these minerals. And many, many papers like this were published in the 1970s. Again, looking at things, measuring things, characterizing things, but not really yet understanding how these biominerals were formed. As you step forward into the 1980s, this is where things start to look more modern, if you will, where people are putting together integrated methods to recognize these complex associations between mineral, crystal, and the biological organic matrix. And you start to see words in the literature like this, where the teeth are forming within an organic matrix of macromolecules. or the mature minerals are forming via some transient intermediate phase. And these are words that we're going to see for the rest of this talk. And then there's a lot of discussion, and there had been all along, but there's more deliberate discussion about structure-function relationships and biomaterials, and biomimetic words start coming out as we think about things like hard surfaces and how they would interact with those tissues. So in this case of the chiton teeth, we have this functional material that serves as a hard part for scraping lichen off of rocks, but it's supported by this pliant, rubbery type of almost like a gasket material to host it and make it more functional than it would be on its own as an individual mineral. So in the 1990s, this is where Trish gets involved, because I was a new professor at Virginia Tech, or excuse me, at Georgia Tech, and I had just done a postdoc at Stanford. And there was this new thing called atomic force microscopy. And as I was a new professor at Georgia Tech, we started to think about how we could connect things like measuring rates of crystal growth and looking at what the consequences for morphology. And the science was so simple when I think back on it, but it was where we were at the time. And as you'll see from my collaboration with Jim DiOrio, who's a crystal growth physicist, we were able to apply a lot of crystal growth theory to it and really start to hooking things together and working with modelers around different parts of the country to try to understand how these, different things like simple amino acids were interacting with steps. And then also, what were the consequences for those signatures, those compositional those impurity levels that you see in biominerals, and can we reproduce that, and what is the explanation for what we see in nature? Remember, those books from the 1800s were produced by all those folks had started to think about this, but how did it actually form mechanistically? So our questions that we were after at the time, or at least we thought we were after, were what are the pathways to formation? And how can we get at that by understanding the systematics of the thermodynamics and the kinetics of crystallization? So again, the AFM was this huge step forward for understanding crystal growth in aqueous solutions. People had thought about crystal growth at ultra high vacuum, but being able to image growth in the presence of water was just an extraordinary change for the community. And here's a picture of my friend Jim. And what they were doing at Lawrence Livermore at the time were growing these massive, and I mean massive, single crystals of KDP for laser applications. And they were trying to understand all the factors that would affect impurity contents. And they deployed this new AFM method to look at some of those things. And they had imported some folks from Alex Chernoff in particular to help them with the crystal growth theory to understand that. And we took those same technologies and these theories and applied it to step propagation or crystal growth in calcium carbonate. And so here's the beginnings of a landscape which I'm going to build out for you today. And first thing I want to talk about just for a couple of minutes is the idea of crystal growth by step propagation. This is what we call classical crystal growth. And we thought we knew the principles, and we thought we understood something about inorganic ions from other kinds of experiments, but to actually image ions, steps, individual steps, growing and moving along, and how their movement and growth rate depends on supersaturation and impurity content was a game changer. And so you're going to find that I like quotes a lot. But so here's another quote. No, I'm going to save that one. I'm going to save this quote. Okay, here we go. All right, so here's classical crystal growth. This is what you see in the textbooks. And this is what we were going on. Everyone was going on it. And the classical step growth model is what's called Burton-Cabrera-Frank theory from the 1950s. And basically the velocity of step motion, this is how is determined by the rate of ions coming out of solution and attaching to these step edges, either at kinks or forming new nucleation sites on the step edge. And you can measure that by AFM, that angstrom scale. And the relationship is just the velocity of the step motion. It's given by some kinetic coefficient, and that's related to the departure from equilibrium. And so we spent quite a bit of time. We had an outstanding setup at Georgia Tech with an instrument and just incredibly quiet conditions. We were able to make these extremely wonderful images. But the thing I want to point out, because it's going to come up later, is that this is a thermodynamic model that assumes that the step edges are fully equilibrated with the solution reservoir. And we're going to see that that has a little bit of a shortcoming for calcite. But I do want to point that out. And I do want to point out that this textbook representation turns out to be limiting. But we're using it. We're going with it and going to explore calcite growth. And we got images like this one, where here's the apex of a growing calcite hillock. Each one of these steps is 3 angstroms. And this is quite old work. You can see it's one of my first PhD students. And I'm not going to take time to tell you how an AFM works, but basically you're rastering over the surface and the resolution is extraordinary. Like I said, each one of these is 3 angstroms, and you could monitor this movement in a solution. So it's a Goldilocks problem. If it moves too slowly, your graduate student will get old before they graduate. If it moves too quickly, the image can't raster and capture it over the time scale of the step motion. Calcite was perfect, not too hot, not too cold. And we did a bunch of experiments with this. And we even did stuff for fun. It has nothing to do with biomineralization. I just want to show you that we could do incredible. Henry did these incredible experiments where he tested for the first time theoretical constructs, such as the Gibbs-Thompson relationship, where you're looking at the birth of new steps at the apex of a hillock. It gives you really nice insights about crystal growth processes. What does this have to do with biomineralization? Well, we were starting to ask this question as well, because there were some shortcomings. And the more we talked to people and read the literature, the more concerned we all got for 3 primary reasons. First of all, calcite and other sparingly soluble materials, because remember, this was developed for the whole theory was developed for soluble salts, don't satisfy the critical assumption that you have that infinite reservoir of kink sites to interact with the reservoir. That's a little bit of a problem. And so as a real result, the steps grew kind of slowly. It would take forever to form a biomineral, but we knew that organisms form calcium carbonate biominerals within a matter of hours. This was just a fundamental problem. Second, we did lots of different kinds of experiments, and the impurity concentrations didn't really match up with anything, but that related to the real world. And we attributed this to vital effects and biological effects. They're certainly important, but it was concerning. And because we know that organisms can grow calcite, for example, with considerable magnesium and other ions as well we played around with. And third, people were really talking about how biominerals X-ray and as single crystals, but the closer you look and the more you look at these samples by TEM and these other methods, realize that they're actually made up of a bunch of co-aligned particles. What the heck is that? That's not at all what we're talking about here. So some of these shortcomings led to us and myself and Steve Weiner and Jim were the leaders of this. We ran this short course in 2003 on biomineralization processes. So the idea was to step away from all these descriptive approaches and look at how biominerals grow. And we got this group of speakers together and we had this wonderful short course. I think there were 115 people from 14 countries, wonderful event, and talked about this. Well, one of the things we figured out putting together the volume was we didn't know much about processes. It was kind of shocking how little we actually knew about how they formed. Lots of descriptions, lots of arm wavings, but what do we really know about it? But one of the things that did come out of it, that's going to come up again later, is Andy Knowles' chapter. If you've ever not looked at this volume, I would tell you the one thing you need to read is Andy Knoll's chapter on biological evolution of minerals. It's just wonderful. And he's written all kinds of books. He put a lot of it together in this particular chapter. And he makes this point that biology is chemistry with a history, right? That makes sense. And another point that came out of this is a lot of discussion. Crystal growth of biominerals can happen by this classical method very often. It's important, it's in there, but there must be more to it, a lot more to it. And so here's a great example where you have a coccolithophora. This is a phytoplankton calcifying. Each one of these platelets these are seen by SEM. Each one of these starts off as a wreath, if you will, of 40 nanometer ROMs of calcite. So they're nucleated and grow to 40 nanometers. And then, as determined by the biochemistry that's local to that setting for that species, they differentiate into different morphologies depending upon the species into something like this. And you'll see this in the book. Prior to this time period, people were drawing coccoliths like this, trying to reconstruct them from a crystallographic point of view. But it wasn't very satisfying when you see this. And when you get in a room full of biologists who are doing all these cryo microscopy methods and just saying, I'm sorry, folks, that's not how it works. And so it was a lively time. It was a wonderful time, I think back on it. So, let's see. I'm using there we go. And so another thing that came out of it after this meeting was Alex Navratsky was attending. I think many of you know Alex Navratsky. And the weekend after the short course, we were at dinner and she goes, I'm really inspired. I'm going to go home and write a paper this weekend. So Alex was, right? And she did. And this is a quote from her paper, because the issue of amorphous phases being having a role in forming these biominerals was coming up over and over again. And I took this from her paper. She published in PNAS in 2004, shortly after a short course. She says the role of amorphous precursors in biomineralization is just beginning to be appreciated, whereas this idea of amorphous intermediates is well known in ceramics and sols and such. I faced this kind of obstacle a number of times in geochemistry, where something is widely accepted, materials physics, but the geologists are not into it. They just have some other closed door way of looking at it. It's kind of interesting way science evolves, isn't it? She published this paper, a lot of discussion happened. I think it opened it was an exciting time. And so where did it go? It led to a lot of people, and we're already thinking about it, don't get me wrong, a lot of people were already starting to think about the idea that biominerals are formed via an amorphous pathway. And so we and others worked on this process, really thinking about what controls, how do you form these amorphous intermediates, and how do they transform, and the beginnings of thinking about non-classical crystallization in this way, right? It's existed for decades, but thinking about it in this way. So let's see. Right, so this came out a little bit later, but I want you to see it because it's so fantastic. Hoopa Gilbert imaged using Zane's very special method to work with these soft materials, the spicules of sea urchin embryos, and she showed their transient evolution as they form as embryos and then transform to calcite. And you can see these color-coded temporal, they're color-coded by phase, where you go from a hydrated amorphous calcium carbonate, and here I'll just point here, hydrated amorphous calcium carbonate to an anhydrous form to calcite. And you can see from 36 to 48 to 72 hours, it's not just calcite, there was a pathway to the calcite. And this reminds me of Dr. Bindy's quote on Tuesday, where he said that because these things can't happen, they didn't. And until someone called it out and said amorphous phases are important, really demonstrated to the community, People didn't see them, and suddenly they're seeing them everywhere. And let me see. And so we did some simple experiments looking at where we made things like amorphous calcium carbonate. And then based on insights from the literature and talking with people and biological systems, we were able to direct it to any of 5 different polymorphs: low mag calcite, high mag calcite, aragonite, monohydrocalcite, that was the most surprising one, and vaterite. And pure forms, not just a bunch of junk mixed all together phases. These are, you could direct, individually tune the polymorphs that form. And one of the things we haven't talked about is organisms do this, right? Form calcite and aragonite in a very controlled process. But here's another problem though. Even though we have these products, they look like they're particulate, right? Look at these particles here. If the classical theories, if you were to go to the classical theories and try to understand this, they would say, oh, these features are just byproducts of surface nucleation and coarsening. Yeah. Okay, well, in cases they are. I'm sure they are in some cases. But what we start to see, we meaning the community, right? This is so much bigger than me. I'm just such a tiny piece in this whole thing. The community started to see more and more evidence in engineered and natural systems of crystals growing by the attachment, formation of particles, and their aggregation and attachment, and in hydrothermal cases, and healing into new single crystals. Great stuff. I would love to have been able to show you. And so this concept of crystallization by particle attachment began to emerge. And here's some pictures. This came from the paper that resulted later, but I just want to show you some examples. These are inorganic systems, all formed in the presence of water, okay? Titanium oxide, here are TEM images of particles that have come together. All of this work belonged to Li Pin. You can see it in the paper, all the citations for this ferrihydrate. We had some of the best images we had at that time were from Li Pin's work. Nanowires. Here's a lead selenium honeycomb material, which is out of my world. Again, a bunch of particles are attaching and co-aligning. You can see that by SEM. In natural systems and biominerals, you can see particles of calcite aggregating and coalescing in sponge spicules, protozoa, aragonite form terraces when there are particles that form these terraces or these blocks. Sea urchin spicules, I've already shown you this. And here's some other pictures of aragonite nanoblocks. So these kind of pictures were previously published and they were coming out, and there was a discussion at the University of Leeds on biological crystallization that I attended. And all these different communities were just talking across each other, mesocrystals, all these different phenomena and observations. And so I came back home and got the Department of Energy to give us some money, and we had another workshop. This time it was a smaller one, and bunch of people came from all many different countries. I've forgotten how many. Let's see. There's well, you can count, but all different expertise, from biologists to physicists, chemists, geologists, everything. And what we came up with, and you just saw 2 of the figures that came out of that paper, was this conceptual model for how crystals grow by this process of particle attachment. And at this meeting, we asked a number of questions, because I remember in the beginning, we were just a jumble of questions and how to even go at this problem from all these different communities. How are we going to solve this in 2 days? Well, we didn't solve it in 2 days. It took like 2 years to work through it and come together with this paper. So what is this? So here are the key questions. What was the growth by a particle attachment? What are the processes? What determines whether it forms classically or by this nucleation aggregation phenomena? And what are the whole factors that control that? The key figure that came out of that is this one that shows multiple pathways to forming a bulk crystal. And here where I started today, I talked about this monomer by monomer, the so-called classical approach. We looked at formation through an amorphous phase and crystallizing to a bulk crystal. But what the community was seeing in all these materials, and I just showed you some of these, are all these other ways that nanoparticles can form and then aggregate, either oriented, semi-organic oriented, sort of amorphous, and then age or not into materials. It also raised the question of, when you end up with a crystal like this, how do you know what its pathway was? And do these different pathways explain some of the offsets and composition that we see between what you would predict by single crystal growth versus the formation of a larger material from many different particles? So this paper, it's one of those things. Once you see it, it's everywhere. And again, because it can happen, now it does happen. And we're seeing a lot of different papers on this area. So now this starts to come around. The very last part of my talk is just a little bit about how the organic matrix plays a role in this. We just had to leave it behind to look at all these other phenomena for crystal growth. So remember, those biominerals are forming in that organic matrix of macromolecules. And how do they control? They obviously do. I don't have bones growing out the side of my head, and we know mollusks reproduce their biomineral structures generation after generation. And so biology has a remarkable control over this. And what are the pathways for whether it's really going to crystallize or form through an amorphous process? And I won't be talking about that today. So I just want to say a few things about this last piece of this landscape, is crystal nucleation. And we did some experiments with surface assembled monolayers, just looking at fractional groups on it's a big organic molecules that are laid out on a substrate, and there's bristling with these functional groups. We've looked at some of the kinetics and thermodynamics of that. And then more recently, we've turned our attention closer to the organic matrix, this tiny baby steps, because these systems are so complicated. Proteins in particular are wildly complicated to work with. And there's been a lot of what we call cook and look studies of how proteins influence mineralization. It turns out that polysaccharides, particularly using the synthesis methods, you can create these tailored derivatives to do testable hypothesis-based studies. And I just want to show you very briefly some of that. And we're getting ready to do some work with peptoids, which are also pretty exciting materials. So again, biology is that chemistry with a history. The answers are here. They're all here in what organisms are doing now and what they have done over geologic time. And so if we look at what biology tells us, we see that these two materials, polysaccharides and proteins, are really important in the organic matrix, of course, in all diverse organisms. And here you see some commonalities on that diagram. Chitin and chitosan showing up here. It's actually everywhere, but this is just what we had at the time. Sulfated glycosaminoglycans, these are disaccharide materials. These are actually everywhere as well. We're just showing some simple examples. Cellulose, pectin, we haven't thought much about these. These different kinds of galactans show up. They are different parts. These are a little bit more narrow in their occurrence. Alginic acid, really important, all fully carboxylated. And the community has traditionally given its attention to two things: proteins and carboxyl groups. And we would argue, and so the carboxyl groups are anionic, negatively charged. We would argue that, yes, those are all important. I think the landscape's broader than that. It's about the functional groups and multiple functional groups that confer negative charge. And this idea that's kind of crept into the community that polysaccharides are just an inert framework. Well, we have a paper that talks about why that's probably not the case and why we got thrown off track about that a little bit. So they present polysaccharides and glycoproteins, which means they have polysaccharides stuck to proteins, present all these different functional groups. Yes, carboxyls are everywhere. Sulfates and phosphates are also prevalent, also conferring negative charge to an interface. And so we want to ask the question here, or how important are these? And I'm just going to say a couple of things about it. And that is that how can we break down all this complexity? How can we break all this complexity down to answer questions about how the organic matrix has structure function, what its structure, function, and relationships are? Here are some of the parameters. It's like a palette of color and the various ways macromolecules can influence mineralization in these kinds of settings. And so conformation, well, that's a tough one. We haven't gotten to that yet. Morphology, yes. Size of the molecule, macromolecule, the composition, and the charge density. The two I wanted to show you just a couple of things. And we're going to do that using sulfated derivatives. And I'm just going to show you this briefly to give you an idea of the palette. You can take things like chitosan now with sustainable biomaterials type research science, and you can sulfate it. You can make your own palette with different amounts of sulfate groups at different positions, whether they're at an amine group or at an oxygen group. You can create a whole palette of materials with different amounts of sulfur and positions and run these experiments to look at how they influence crystal formation, calcite formation. And we do this, Kevin, Edgar, we do this, and Brenna and I, we all work together on this project. It's just published. We're also working with heparin molecules, amazing molecules that are prevalent everywhere throughout the phylogenetic tree. They have lots of levers to turn in terms of both amine carboxyl groups that are present and multiple regiopositions for putting sulfates on there or carboxyls for that matter. Really interesting molecules biomedically as well, because this is what you get at the hospital if you need a blood thinner after surgery or if you have some kind of cardiac condition. So fascinating molecules. So we're doing a lot of experiments with these sorts of things. And I'm just going to show you the punchline. Here's the interfacial energy barrier to nucleating calcite onto these materials as a function of their net charge or the monosaccharide. We've done modeling studies as well as calorimetry studies to try to understand why this happens. But you see this general trend here with the change in the energy barrier with increasing charge. And it includes both carboxyls and sulfate groups arguing for the generality of these molecules. In the modeling, we've shown that there are all these interactions, the calcium with the macromolecule are solvent separated, which brings up some other interesting questions about what's really happening at these interfaces. And I just want to leave you with the idea that functional group properties, not molecular class itself, it's the local phenomena that are influencing crystallization. There's more I could say about this, a lot more, but I will spare you today. So here's our landscape that we put together. And as we look at it, gosh, I never intended for it to look like this. You just start out as a new professor and you just don't know where science is going to take you. And it's a community effort, right? So many people have come together. There's a curious thing here though, because crystal nucleation in itself does not make a biomineral. We're lacking in connection through, are those nuclei attaching or are they undergoing step propagation to form? Is it a non-classical crystallization process? There's so many white spaces on this diagram. It looks like it's finished. It's just not even started. So if you look ahead to some of the things that you might ask, and some of the big things, there's lots of big things actually. What are the details of how those particles interact? What is happening at the solvent with the solvents, ions, or the water molecules, and the ions themselves to influence how these particles organize themselves. And you can look at TEM movies and see how the particles are touching and attaching, detaching, until they find, if you will, the most thermodynamically stable place, and then the attachment occurs. It's really quite remarkable. It's surely some combination of kinetic and thermodynamic drivers. And then I show this to my students sometime. This is what we're after. This comes from one of Bob Hazen's papers, right? This is this landscape. It's n-dimensional. Rather than having a palette, it's this n-dimensional landscape of the different parameters that make some kind of function possible. Are they tolerant? Is the function possible over some range of parameters? Yeah, in some case, probably. Is a big range possible and you don't have optimal function? Absolutely. Is something very special possible with all those in parameters that give it extraordinary outcomes? You get the idea. Spatial, temporal, and chemical combination of things. What are those things? We don't know. All right, so wrapping up, I just give you some parting thoughts for those of you just in the middle or starting your careers. And that is three things. Two. First, the literature is full of clue cards. And this is something we say in my group. We have the clue card because we saw this thing in the paper somewhere. Gives you something to think about that's, might not have been commonly accepted at the time. They might have been denied tenure for it back 40 years ago. And I know of examples of this. But they were a clue card. It tells you something important. And actually, when I was a postdoc at Stanford and I was going to get into crystal growth, somebody told me, will not remain unnamed, everything about crystal growth is already known. This is in 1993. Other, characterize your materials. Everybody knows this. But, you just have to be open to new ideas. And somebody else said, was pretty aggressively, there's no such thing as amorphous calcium carbonate, they said. And okay, sure. All right, we'll see. And finally, because I love quotes so much, look for connections, thermo rules. I was at the Goldschmidt conference in 2005 and Alex got up and you can see she's one of my mentors and my treasured people over the course of my career. She got up and gave her Urey Medal speech. And she said, and I wrote it on the back on a business card in the back of this, my name text. I had nothing else. Ideas developed in one context become important, even crucial for other applications, sometimes much later. And I love this quote. It applies to everything, that we're all working on and looking ahead to where science is going and all the exciting questions that we need to be asking. All right, so I think that's it, and I appreciate your attention. Thank you very much.
Thank you very much. That was fascinating. I have no background knowledge of any of this. So would we have any questions for Patricia? Yes, excellent. Hold on one second.
Do I really need a mic?
Well, for the people online.
Thank you very much, Trish. First, congratulations on being the IMA medalist. This was
Thank you.
Of 2022. You talked about different pathways from going from solution to final crystals. I'm intrigued to know whether the organic molecules play a role at every stage, and, for example, would maybe control the structure of the amorphous materials that then subsequently crystallize. So what step is the polymorph? I'm a crystallographer. I'm interested in what step is the polymorph of the product, the crystalline calcium carbonate product, determined, and by what mechanism?
Oh, here, sorry. Okay, sorry. Yeah, so I have to confess to not being able to hear most of the question, Ross. You and I have the same problem, okay? But the short answer is macromolecules are playing a role at the very beginning, at every stage, right? And so you can see this in some of the studies with proteins, and we can't do that with our molecules, but just the configuration of the compartment and the local space is going to play a role. One of the interesting things that we've been thinking about is how the ions for calcium carbonate precipitation are not binding directly to the macromolecules. And we can see this through the calorimetry studies, that the free energy of the interaction is really low, just like what you see in biological thermodynamics, right? All of those reactions are very low energy, same kind of thing. And so much of the system is solvent dominated. And I, of course, didn't talk about any of that, but solvation really rules here. And so, and the local environment of that water is going, of course, going to be important.
We should have time for one more if anyone has another question. Or nothing. All right, so I have one that is probably very amateur because this is very far out of my field. From the geology point of view, the next question is often, what can we do with this information? What are the applications? Can we?
So the applications are many, right? If you look at the material science, and I have an appointment in the material science department, people are forming or trying to get insights from the very beginning of this, from the 80s, right? From the moment you heard biomimetic materials, people are trying to apply this to form new materials for sustainable technologies, for new types of energy applications, you know, batteries and all these things. Biomedical is a huge one, and drug delivery, that's a particularly big one that our group interacts with. Environmental applications, they are numerous. And so some of the papers we published recently, we talk about this quite a bit because there are many.
Thank you very much. Okay, we will see Patricia again tomorrow for the medal ceremony. Thank you very much.